All right, guys, so in this FRQ, I'm going to be going over numbers 1 and 3 on the assignments for this week. So this first one is going to be number 1. So let's read through it and see what they're asking about and how you would get maximum points on this one. So this one talks about researchers conducted a study to investigate the effect of exercise on the release of prolactin into the blood. The researchers measured the concentration of prolactin in the blood of 8 adult males before T = 0 and after 1 hour where T = 1 of vigorous exercising. So after this, as a control, the researchers measured the concentration of blood prolactin in the same group of individuals at the same times of day 1 week later, but without having them exercise. The results are shown in figure 1. So basically we have it with exercise and without. And so at T = 0, you can see that they're measuring in nanomolars per liter. So in terms of this, it looks like there's not a difference in the prolactin levels because the error bars overlap. And this is with exercise. And then they measured it a week later of the control group at T = 0 without exercise. So so that's kind of what they were doing on that experiment. So let's kind of take a look and see what the question is based on. This one says prolactin is a peptide hormone primarily secreted by the anterior pituitary gland. It performs over 300 functions in the body, mostly related to metabolism, reproduction, and regulation. Based on its structure, describe the most likely role prolactin plays in a cell communication pathway. Explain why this is most likely the role. Okay, so gives you a little bit of a backstory on this, what prolactin is. You have to figure out what does prolactin play in the cell signaling pathway and explain why. So key evidences here on this to get some points is that that signal, it does many functions. It can increase metabolism. It's going to help increase reproduction. It's going to use for regulation. So another key word that it tells you here, if you didn't catch it, it's a peptide hormone. So that tells you it's a protein. Proteins for the most part, like as a whole, not talking about 1 or 2 amino acids, but proteins as a whole are a polar molecule. And it even tells you it's a hormone. So that kind of leads into cell signaling of where this thing would fall. So on the left, we got paracrine signaling where signals don't travel very far. On the right, this is from chapter 11. This is showing an endocrine cell. Each one of these blue dots represents the signal. And that essentially would be where that prolactin would be. So basically, if you forgot, remember that prolactin is going to search and go and travel through the bloodstream if it's endocrine signaling. And it's going to try to find the cell if it has the right receptor protein. Okay. Now, just kind of give you a brief overview while we're here. Remind you guys of cell signaling, right? This would essentially be the polar signal over here on the left, binds to the receptor protein. In the transduction phase, this is where your signal gets amplified. The majority of the time, the signal is going to activate these proteins, these kinases. This is going to activate that one, activate that one, activate that one, activate this one, to eventually activate what's called a transcription factor. Now, if you forgot what transcription factors are, go back and watch the chapter 18 videos. But basically, it's a protein that just binds to the DNA and it turns on the expression of that gene. So that means you're actually going to read it. So in this case, the signal activates the transcription factor, which is going to go make mRNA, and this is going to go and make a protein to do a specific job. So if you're thinking on this one for this FRQ, basically prolactin is going to tell a cell to make enzymes to do metabolism. So breaking things down, making cells do reproduction, making them do regulation. So all these are pretty broad terms, but basically you want to know that if we put this into context, that if this is prolactin, that's the signal. It can activate various functions, and that's kind of the key thing on here. This is just another thing, another bit visual showing you that you get your signal. You get these. Remember that these are kinases in the transduction phase. This activates that, activates that. I just wanted to remind you again, if this is your reception, then this is your transduction phase. Remember that in the transduction phase, it gets amplified, right? So this one signal can activate maybe 3 of these proteins. They can activate 10, these can activate 20. So this is the process where your signal most definitely is going to get amplified. So just wanted to put that out there and just remind you guys, and now we can kind of answer this. Describe the most likely role prolactin plays in cell communication. The role for prolactin would be that it is going to be the signal. Explain why this is the most likely role. Well, the signal, there's a couple things you could say here. The signal is a hormone, okay, and travels through the bloodstream. And you could also, key things here is that it can activate multiple responses. So that's kind of how I would answer this question, is that you identify that it's a signal. Why is it most likely the role? Well, it's the signal that can actually activate more than one response, and it's sent out via the bloodstream. So that's something that I would kind of make sure you had in your answer. So cool, nice little recap on cell signaling. So let's take a look now at this next one. For part B, state the null hypothesis for the experiment, justify the use of the without exercise treatment as a control group, identify one limitation of the study. So the null for this one, again, your variable is, you're measuring prolactin, but your variable is exercise. So I would rephrase this for my null as that exercise has no impact on prolactin levels. In the blood. All right. Now, justify the use of without exercise treatment as a control. So this one for the justify, the justification of this would be that there is no variable used, okay? And that's essentially measuring prolactin levels underneath a normal, what are normal prolactin levels, all right? Now, for this one on the limitation, there's a couple of things that you could say here that I would think would earn you points on this one. One thing is that there's only 8 males, and they told you that in the problem. And another thing that I would suggest that could be a limitation is that they only measured after 1 hour. Whereas I feel like that one thing that they could increase in terms of getting better data is having more data inputs by having more people in the study. The second thing would be taking data or in terms of prolactin concentrations maybe every 5 minutes. That would be something that I think would be another limitation. So I think those would be the 2 big key limitations on this study. Part C. Part C is using evidence from the treatments. Describe the effect exercise have on the release of prolactin. Justify your answer. Explain what this means in relation to the null hypothesis. Okay, so this one you have to look at the error bars. So with exercise, I would say that describe the effects, the release of prolactin. So if you look at with exercise and even without exercise, notice that the error bars are all essentially going to be overlapping within one another. So I would say that prolactin, or I should say exercise, has no impact on prolactin levels. Justify, well, your standard error of the mean. And again, this is all going to be the standard error of the mean at 95% confidence. So the standard error of the mean overlaps. And that's really when you compare all the sets of data. So to explain what this means in relation to the null hypothesis, again, your null was that exercise has no impact on prolactin levels on the blood. Your data that you collected, you essentially would fail to reject the null. Because those error bars are overlapping, you can't say statistically that they're different enough, that those values were different. So those were those 3. And then for part D, kind of takes another weird, interesting turn. So it says during milk production and secretion, mammals exhibit positive feedback. The hormones prolactin and oxytocin are involved in milk production and milk ejection, respectively. Describe positive feedback and explain how these hormones most likely play a role in this type of feedback mechanism. So kind of a different problem now, but I just want to revisit what positive and negative feedback is. So if you look here on the left, this is an example of negative feedback. So if you look here, if let's say you have a stimulus, and let's say we're talking about blood sugar, right? So if your blood sugar level gets high, you're going to secrete a hormone to lower the blood sugar. So basically, whatever your stimulus is to maintain homeostasis, you're going to release something to bring that back down, or vice versa. Let's say if your blood sugar level gets too low, you're going to release a hormone to help raise it back up. That would be an example of negative feedback. So whatever your stimulus is, you're basically secreting a hormone to reverse it. Positive feedback is a little bit different, where if you release a stimulus, that stimulus that gets secreted, or whatever that stimulus is, you're going to release a hormone to actually increase that stimulus. So positive feedback mechanisms are pretty rare. And there's not as many examples of positive feedback mechanisms because typically it's not about maintaining homeostasis. Usually there's the stimulus and you're wanting more of it. So in terms of this, like if fruit, there's that ethylene gas, we talked about it before, that as these fruits, it release that hormone, that ethylene gas, it secretes that hormone, and it basically promotes other of those fruits to secrete that ethylene gas. So that's an example of positive feedback. Stimulus, and then there's more of that stimulus produced as a result. So in this case, this one was dealing with prolactin and oxytocin. And basically it just wants you to describe what positive feedback is and explain how these hormones likely play a result in this type of feedback mechanism. So this is just showing you exactly what it is. It's basically showing you that the more that the baby's suckling on the breast, basically what's happening is that the more that the baby is providing this stimulus, the more the prolactin and oxytocin are going to be secreted. So this is basically showing you that the more that stimulus is provided, the more that these two hormones are going to get secreted. So basically how I would explain and describe positive feedback, if you're going to be increasing the stimulus, basically you're going to be increasing the response or secretion of the hormones. And so when you think about it in terms of the breastfeeding, the more that the baby is actually feeding, the more milk that is going to actually be produced. So he's doing the stimulus, he or she is doing the stimulus. You're providing those hormones being secreted. And so that's an example of positive feedback. So explain how these hormones most likely play a role in this type of feedback mechanism. I would just explain here in terms of this one what I was already mentioning. The more prolactin released, the more oxytocin released, and the more milk production. So basically, you don't have to get that specific. It just wants you to explain what positive feedback is. So the more one signal is secreted, the more another is going to be secreted to actually increase that response. So that's part D. Now let's move on to this next one. This one is going to be a pretty short one. This is a good one. It's pretty simple on cell function based off of the organelles that they have. So this one tells us that, and this one was number 3, by the way. The following data were collected by observing subcellular structures of 3 different types of eukaryotic cells. So we have cell X, cell Y, and cell Z. So it gives us some organelles. We got smooth ER, rough ER, mitochondria, cilia, and Golgi bodies. So, excuse me. Assuming that these are human cells, identify the likely function of cell Y. So if you look at cell Y, it has a large amount of smooth ER, a large amount of rough ER, moderate amount of mitochondria, a moderate, no cilia, Golgi bodies, there's a large amount as well. So the key thing that I would focus on is that there's a large amount of your rough ER and a large amount of the Golgi body. So you just got to kind of remember, well, what is the role of those two, and what is the relationship between them? So remember that the rough ER is going to be making a lot of proteins, and the Golgi body is going to help modify and package those proteins. So assuming these cells are human cells, identify the function of cell Y. So the function I would say for this one would be protein synthesis. And explain how the data supports this. I would say that there's going to be a high concentration of the rough ER and a high concentration of the Golgi bodies. So just kind of reiterates what I was saying. Those are two cells that are going to be used in the endomembrane system, right? You're shipping proteins out of the cell. Now for part C, predict what would happen to a cell X if the mitochondria were suddenly poisoned. So let's look here at cell X. Looks like they have a smooth ER small amount, rough ER small amount, but there's a large amount, according to these three, of mitochondria. But the other key thing here is that there's cilia. Remember that cilia is used for movement. So what would happen if the mitochondria would suddenly be poisoned? I would essentially say that there's going to be stop moving or even death. Provide reason to justify your prediction part C. Well, no ATP is produced, no movement can happen. So that's how I would justify that. So pretty standard type of AP bio level question. You would have to analyze everything looking at the data. So anyway, so those were FRQs number 1 and 3. And we'll see you guys in the next one.